So it's been a couple months since my last update, so we're overdue. And I decided to go ahead and give another update on our progress in developing treatments for RVCL. So for anyone who's unfamiliar with the disease and uh, with what we're trying to achieve here, RVCL stands for retinal vasculopathy with cerebral leukoencephalopathy. It's a mouthful. Uh, um, the short version is this is a genetic disease. It's inherited, and it's inherited in a dominant pattern, which means uh, each person in an affected family has a 50% chance of inheriting the disease-causing mutation from an affected parent. And patients with RVCL are typically healthy until they're anywhere between their mid 30s into their 50s, and then they start to develop symptoms and organ damage. In all patients with RVCL, multiple organs are affected, including the brain, the eye, the liver, the kidney. And so we are uh, working to develop a treatment. So my name is Dr. Jonathan Miner. I am an associate professor at the University of Pennsylvania Perelman School of Medicine. I moved here 3 years ago. When I moved, we did not have any prospects yet for therapy. Now we've developed a gene therapy, but we have to prove that it will work in people. More on that in a moment. There's a second avenue that we're developing, which is small molecule drugs. So small molecule drugs are drugs that can be taken as a pill. They'll go into every cell in the body and will, in this case, we hope, halt disease or slow its progression. So the reason this disease happens is the abnormal gene encodes an abnormal protein. Proteins are the machinery that make us work. And in the case of RVCL, the abnormal protein is toxic. The abnormal protein in patients with RVCL starts to attack the chromosomes within the cells in the body. And it does this, as we found, more with age. And that's why patients with RVCL go from being completely healthy to having a deadly disease uh, uh, sometime between the late 30s and 50s, usually. Um, and so what we need to do is we need to stop the abnormal protein from causing organ damage. And one way to do that is a small molecule, a drug that can be taken as a pill. It would go throughout the body, attach itself to the abnormal protein, and prevent it from causing damage. So about a year and a half ago, we had identified a compound that was generated by our collaborators at the University of Michigan, and we found that that compound was protective. But there were problems with it, and I can't go into too many details about the compounds and what all the different problems were, but it would protect cells growing in addition in the lab, but it was not good enough to protect an animal. So a few months ago, several patients and I and representatives of the Clayco Foundation took a trip to Michigan, and we met our chemist collaborators there. And within just a few months, we've gone from having one compound to having 15 compounds that are effective in addition in the lab. And we're to the point where we're starting to be able to figure out that some of them are likely to be helpful in animals, in humans. And so now we don't want to overpromise. So to be clear, until a clinical trial is done, until we have more data in model systems, we can't be definitive about this. What I can say is the progress is very, very rapid. We've gone from one compound to 15 compounds. Every other week, we're getting new compounds. We're screening them here at Penn. And we have a high degree of confidence that we're going to have a treatment to be tested for patients with RVCL. But there could be, along the way, problems that arise unexpectedly. So it's important for everyone to remember that it's hard to guarantee what will happen. We don't know the future. We can't predict exactly what the outcomes will be in a clinical trial. That's the reason you have to do a clinical trial, is to prove that it works. But what's different now, and I want to emphasize that what's happening now feels very different from what happened in the past. What's happening now is that the therapies that are being developed are custom developed for patients with RVCL. And because they are personalized, because this is a disease where there is only one gene that's abnormal, and therefore one protein that's abnormal encoded by that gene, because there's one target, there is no reason why we should not be able to design a custom drug for this disease. Do everything we can to make that accessible to the patients. So we're working day and night, weekends, late at night, working like crazy to get this done. And we're committed to that. The important thing to know is that there is a path toward curing this disease through hard work and science, but the support of the patients, the families, Clayco Foundation has been instrumental in all of this. Without the support, none of this would have been achieved. But we're not done yet. There's a long way to go. And going from having a compound that works in the laboratory to having a pill that a patient can take is still a long, hard road. We need to get this done as soon as possible. Now, the other side of this is the gene therapy. So we have developed gene therapies for patients with RVCL. We started with the most common mutation. We're almost to the point where we have a gene therapy for every family with RVCL. So this is a little bit of a different situation. So small molecule drugs would be taken as pills. Those would be protective no matter what kind of mutation a patient has. So the concept is this: within the abnormal gene in patients with RVCL, different families can have different mutations. The small molecule drugs that would be taken as a pill that go into all the cells in the body and interfere with the protein, that would be protective, we predict, for any patient in any family. But the gene therapies are different because different families have slightly different mutations. They all cause a similar abnormal protein. They all cause the same disease state. But because the DNA sequence can be slightly different from family to family, what this means is that one gene therapy would not necessarily be right for every single family because there's a different DNA sequence in the different families. So another layer of complexity. So there's a new technology that we have implemented that can correct the disease-causing mutation. We started with the most common one. We were first able to demonstrate we can correct the mutation in a dish in the lab. And now we've demonstrated that we can correct the mutation in mice that we've engineered to have a human TREX1 gene. So these are mice that we made as soon as I moved to Penn. They were designed specifically to test small molecule drugs, which are the drugs that block the abnormal protein, and also to test gene therapy. So next, in cells growing in dish in the lab, we have engineered all the different families' mutations. And we are almost finished making a gene therapy for each and every family. So what does this mean? So a gene therapy would be a drug that could be given, for example, intravenously, that would go into the blood vessel wall, enter the cells, and change the abnormal gene into the normal gene. The important concept here is to understand that we cannot guarantee this would be a cure because we don't know what percent of cells need to be corrected. There's still a path that needs to be traveled in terms of understanding what would be necessary. But this field is moving forward exponentially. We are getting better and better at this. And we've already proven that we can fix the abnormal gene, the human gene introduced into an animal model. We can turn it into the normal gene in the liver. Now we're working on doing it in different organs, in the blood vessel wall. we have a very high degree of confidence that this could be an effective treatment for RVCL, and it might even be a cure. It might even be a cure. So now you have to sort of step back and think about these two paths. One, a pill. The pill would go into all the cells in the body, block the abnormal protein, and limit progression of disease. That's for any patient with RVCL. The gene therapy would be something that has to be custom designed for each family, that could be given repeatedly. And every time it goes into the body, it would, in a certain percentage of cells, change the abnormal gene into the normal gene. Both of these things are real avenues for treatment and potentially a cure. And so the future is bright. We're excited about this. We want to go fast. We know that there's, the time is of the essence, and we don't want to let people down. And so I also want to emphasize that we can't predict the timing of when this will become available. That is completely dependent on how hard we work, whether there's something unexpected that comes up along the way, which is something we can't control. We can't control how hard we work. The resources that have been donated to Penn, the Clayco Foundation, have made a huge difference. We wouldn't be where we are without the support that we've gotten. When patients reach out to us, let us know what's going on with them, let us know about their family's mutation. It helps us ensure that we're covering all the families. I don't want to overpromise because it could be that we would pursue a clinical trial and there would be a side effect, for example. But so far, we don't have any reason to doubt that we're going to get this done. And so anyone who has any questions or concerns, please feel free to reach out to me anytime at all. We're here to talk. We want to keep the public, the families, the patients informed about everything that's going on. And I want to assure people that we are regarding this problem, this disease, as if it is a problem that affects us or our own families. We're taking it that seriously. And so please feel free to reach out anytime if you'd like to get more information, and I will make myself available to talk. Have a nice day.